Hello, welcome back to our last installment of cell signaling and cell communication. So here we are back to our original picture. And so let's take a look and see if we can track where we've been. So our growth factor blob here was our step one. That was cell signaling, sending the signal. Our purple receptor embedded in the plasma membrane, that was our step two, which was signal reception. All of these arrows are going to be step three, which is our transduction pathway. So things are happening. We saw different kinds of examples of transduction pathway. And it looks like, I'm going to say, I don't really see a product too much, other than we're probably going to get some kind of protein. It looks like some protein synthesis is happening down here. So I'm going to say that is our step four. So that's the result of whatever, I don't even know what pathway this is, but it looks cool. So this is our four steps. Let's take a look if we can track that in our diagram. So we've covered signal transmission. So that was neurotransmitter, hormone, paracrine, and direct contact. We had reception, which we had ion channel linked receptors, G protein linked receptors, enzyme linked receptors, and intracellular receptors. For signal transduction, we had open closed channels, phosphorylation cascades, second messengers, and for the intracellular receptors, it was gene expression, so transcription translation. And then the response. Yay, we finally made it to the end, the last stage in cell signaling and cell communication. Let's take a look. Response to signals. There's a lot of responses, but I don't have a lot of slides. This is going to be a pretty short video because I'm just putting the cellular responses in kind of these general categories because it would just be insane to try to list all the examples. So the first type of cellular response that I have listed here is membrane permeability altered. So I just put up a cool picture of two cells next to each other. You can see those nice dark two lines, which is the phospholipid bilayer. So this would be the phospholipid bilayer of cell 2, and here's the phospholipid bilayer of cell 1. So we saw in our ion channel linked receptor where the neurotransmitter came in, bound to the receptor, opened the channel, sodium rushed in. You've just altered membrane permeability. That's a cellular response. That's enough to sometimes activate a target cell. It's how your neurons are signaled to fire. It's how your muscles are signaled to contract by altering those membrane permeabilities, opening ion channels, letting ions flow in or letting ions flow out. Okay. The next one we talked about was enzyme activity was altered. We saw lots of examples of this. So I just put a picture of an enzyme and its substrate with its active site and allosteric bind, all this stuff from chapter 7. So enzymes altered, we saw kinases activate other things by phosphorylating them. We saw the enzymes associated with the G protein linked receptors activating their pathways. And just in general, enzymes are turned on, or maybe enzymes are turned off, either by the DAG pathway, the calcium pathway, the cyclic AMP pathway. Ions coming in could trigger enzymes. So enzymes being altered is a cellular response. So many different examples. And then the last one that we just looked at with intracellular transduction pathways is gene expression. How are genes regulated? Are they turned on to express and make their protein product? Are they kind of tamped down a little bit to not produce their protein product? So this is a nice review of our chapter 13 material where your instructions are encoded for in the DNA, the master blueprint. We have a temporary short piece of RNA that's going to code for just the gene. That's our mRNA. The mRNA goes out and lands on a ribosome for the process of translation. tRNAs are involved. They bring in the proper amino acid, and then our product is a polypeptide chain. That's how the cell can respond, is by pumping out whatever that protein product is that the gene is encoding for. It could be an enzyme. It could be a structural protein. It could be an ion channel. It could be a proton pump. It could be, whatever. There's a ton of things I could just keep listing. So these are the three major categories of cellular responses: changing membrane permeability, regulating gene activity, and regulating enzyme activity. And all of these things could be up or down. So you could open channels or closed channels. You could turn on enzymes or you could turn off enzymes. You can turn on genes or turn off genes. So it goes in both directions. It doesn't always have to be turning something on or turning something off. All right, so that's it. Those are our 3 cellular responses. So that is general categories of that step. So what I wanted to do, this is out of your textbook. This is a real cell transduction pathway. It's called the RAS pathway, which is common in certain cancers. The cancerous part is what happens is this part is damaged. And so this enzyme never goes off. And that's the bad part of it. So it is because of a mutation, the tyrosine kinase enzyme is mutated. It turns on even without a signaling molecule coming in this case, epidermal growth factor. That tyrosine kinase is always on. And so this pathway is always on, always on, always on, causing the cell to reproduce, and that's our cancer. We saw that back in mitosis. So this is that's how it was identified, this RAS pathway. It helps to regulate some of the proteins involved in cell division, which is kind of cool. So you don't have to memorize this, so don't think that you have to memorize this, but I just wanted you to see a various combination, because things don't always have to go exactly the way that we learned them. Because what do we have? We have our signaling molecule, our little sunflower seed. The receptor is a tyrosine kinase receptor, but it is involved with a G protein. So it's a tyrosine kinase and enzyme linked receptor that's also linked to a G protein, right? So here's your G protein that's going to activate this RAS enzyme, okay? And so then that's going to trigger a phosphorylation cascade, which is going to lead to an end product, which is a transcription factor regulating gene expression. So I think I've just mashed like 5 different pathways that you learned differently into this one pathway. Now, I didn't make it up, I'm just sharing it with you. But this is an example to show you that you can have a combination of all of those things. So you can have a G protein linked receptor go to a phosphorylation cascade, opening up an ion channel. You can have an ion linked receptor allowing ions to flow in that could trigger a G protein that could trigger a transcription factor. It's like a mix and match. So I just want you to remember things as I describe them in those different sections, in those little videos, not so much all the different permutations that can happen. Just know that that's possible, but I'm not going to be asking you to mix and match for any particular reason. Okay, so then the last thing is just to realize the power of these signaling molecules. So let's say you have one receptor, one G protein linked receptor, one little radiator, right? One little radiator. But that one radiator might have multiple G proteins, many G protein molecules, right? Look at all our boomerangs we have. So one ligand combined to one receptor, which could activate 4 or 5 adenylate cyclases, which each one of them can convert ATP into hundreds or thousands of molecules of cyclic AMP, which can then activate hundreds of thousands or more kinases, these phosphorylated proteins, which can then, it's like a divergent pathway. So one hormone, say epinephrine or insulin or whatever the ligand happens to be, just one could result in this huge signal amplified inside of the cell. And that's one of the ways the cells can be very efficient. If we need a quick response, just increase those different parts of this transduction pathway and you'll get a huge response. Instead of having to stimulate the cell 50 times to get the same amount, you can stimulate the cell 1 time and get a huge result, a huge response because of the signal amplification. Just depends on what the cell has going to make it sensitive, or the ability to activate maybe 5 G proteins instead of just 1. All right, so that wraps it up. That is our last lecture, you guys. So I've enjoyed this term. I hope you've enjoyed it in the weird way that we had to do by 211. I'm hoping that we'll be back on campus soon. I know summer is online. I might see some of you in other classes this summer, or maybe in the fall if you do 231 or microbiology. I can't promise whether we're going to be on campus or not. We'll do the best we can with what we have, just like we did this term. All right, thank you so much, and I will see you next time, hopefully, maybe. Okay, bye.